In our final module here, we're going to look a little bit at nucleic acid, so DNA and RNA, and the relationship to protein synthesis. This could easily be its own module. This could easily be its own, field of study practically. But we're just going to touch here how nucleic acids work into the whole thing. Nucleic acids, DNA, this is responsible for our genetic code. So the instructions for creating proteins and, sort of how we are. And here's the structure of a nucleic acid monomer. We have a phosphate, a deoxyribose sugar, and then a base, which is the part that's going to change in this example. And for this base, we have 4 different bases. We have 2 classified as the pyrimidines, so cytosine and thymine, C and T for DNA. And we have 2 that we'll call purines, which are A and G, and these go together as well, right? C and T go together. A and G go together. We'll see a little bit how on the next slide, but these 4 different bases provide the structural diversity that's needed for DNA. DNA has a really characteristic shape, and this is one maybe you've seen before. On the left, we show this DNA double helix, which is characteristic of the DNA molecule. It keeps the information stored inside, and that information is the sequence of these bases. And when we have this double ladder that closes up, we see that thymine and adenine, right, these 2 go together. A and T, these pair together, and cytosine and guanine, these 2 pair together. So it is this pairing that creates a big part of how DNA is how it is, right? And they pair based on hydrogen bonding, 2 hydrogen bonds between A and T, 3 between C and G. And so, right, this is all going a little bit fast, but this is the basic structure of DNA, and it's this sequence of the bases that codes for what protein gets synthesized. It's the protein that's synthesized that creates all the advanced function that we enjoy as human beings. And so here's an example of a chart that shows how DNA leads to proteins. This chart shows that for a triplet, aka a codon, 3 bases on DNA, it will eventually create a certain amino acid. So if we have a long chain of DNA with a ton of bases, that corresponds to a sequence of amino acids. And so let's go ahead and pause here and try and use this chart. If we have the following DNA code, and I've separated it into 3-letter codons for you, what amino acid sequence is this? And so as we look to the first one, ACT, we can say, well, the first base is A, and so that goes in the 3rd row here. Second is C, so that's the 2nd column. And then of those, there may be some different amino acids that are created. But note that there's also redundancy. So if we miss one of these, we may end up with the same amino acid. Go ahead and pause, and we will discuss. All right, so using this chart, we can see the amino acid sequence should be as follows. And again, right, say that first codon was actually malfunctioning. Say it was ACA. We still get the same amino acids. So we have 64 combinations for our codons, but only 20 amino acids. This creates some redundancy in the system, and that's really important for resilience of the genetic code and, is the way the body functions. And the body has been very successful at evolving many different creatures, and humans, the most notable example of one that's very, very successful here on planet Earth. Finally, right, this doesn't all happen in one place. We carry the information away from the nucleus where the DNA is safeguarded. The mRNA carries the message from the DNA to the ribosome, so that's out of the nucleus into the cell. There's more space and more ability to create the proteins there and then eventually export them. tRNA is responsible as well for bringing the correct amino acid to match the code, and this is the central dogma of biology. Now, for this course, this isn't a process that you need to know every single step about, but the idea that DNA contains the information, it's held in the nucleus. We have other nucleic acids, ribonucleic acids, that are responsible for facilitating this process of getting a protein from the nucleic acid safely. That, I think, is the broad strokes of what you need. And for more information, see the extra credit on the central dogma that's part of this course. I think it's a very fulfilling thing to learn and is a really great takeaway that'll inform your studies as you continue forward.